Thank you, Lucilla, and I'm delighted to be back as part of the iDASH family for round 2. How do we make things go forward? Oh. Got it. Okay, great. So I'm going to be talking this morning with Jihoon Kim because we're a team. Chisato Shimizu from our team is also here. And as Lucilla said, I do wear many hats. I am chair of the pediatric IRB. UCSD runs 6 different IRB committees that oversee our human subjects research. So I would also be happy to answer questions about how we function to protect the children on whom we do research. I've been invited back to iDASH to work on a project that's going to again involve Kawasaki disease, but in a very different way. So we're using this as a model system to get at the issue of how we can share data not only between institutions, but between countries. So I work on a rare disease, and that's really, I think, the nexus of why we need to develop all of this technology for privacy, because we have to collaborate with other institutions. We really cannot do, when we work on a rare disease, we cannot do our work within a single institution because we just don't have the numbers, and particularly when we're doing genetics research, we're going to need to access data from multiple sites and include multiple international sites, and therefore deal with the privacy rules and regulations of those countries, let alone those institutions. So my disease is an acute self-limited illness of children. The etiology is still unknown, although we might have some interesting conversations about that over dinner. We care about it because it's an important cause of acquired heart disease in children, and 25% of children who are not treated in a timely fashion will develop potentially fatal heart complications. And we recognize the disease through the clinical signs that you see here. In the second half of the iDASH life of this grant, we're going to be participating in the 4th biologic demonstration project, and we have 3 specific aims here. I'm really going to be talking about the first aim, which is how we're going to collect DNA samples on African American children and why we think that's a really important thing to do in terms of understanding the genetic underpinnings of this disease and the increased susceptibility among African American children. And then Jihoon is going to be talking about how we're going to be able to share data across institutions and countries and some of the analytic issues in working with these data sets. So I want to make it really clear that although Kawasaki disease was used as a model system in the earlier part of iDASH, there we were really focusing on sequencing and developing pipelines and shareable tools that could help analyze the, in that case, microRNA sequence. The cloud supplement that was funded by NIH is involved integrally with the analysis of whole genome sequence, also on an African American family of Kawasaki disease, and you'll be hearing more about that. And I'll be talking about the wrap-up, the very successful wrap-up of DBP1 tomorrow. And today, we're going to talk about this issue of collecting DNA on African Americans, using sources from other countries to pull together all the data and be able to share it with the multi-party computation and distributed computing. So we're going to test the hypothesis that there's a unique set of genetic variants among African Americans that contribute to their increased susceptibility to Kawasaki disease. We're working with Emory University, which does not have a Kawasaki disease research center, although they take care of the largest number of Kawasaki disease patients in the United States every year. In their system, they have about 130 new KD patients every year, and yet there's no infrastructure at all for collecting data, and we're going to fix that for them. And they're very excited about being a part of iDASH. We're going to create a cohort of samples that are collected on African Americans, but the genetic research supports the idea that we can also pool our samples with Afro-Caribbean samples that were collected by our collaborators in France. So again, rare disease, hard to collect these samples. We need to be able to pool data with other investigators around the world also working on this disease. Jihoon's going to talk about how we're going to do some of the analysis, and then we're going to use as a control group a large data set of genotyped continental Africans that have been collected as part of a different project by our collaborators in London. And again, all of these raise privacy and sharing issues. So these are the data that support the idea that African Americans are overrepresented among our populations of Kawasaki disease patients. There has been no publication on African American KD subjects since 2003. We aim to change that. This really is an ignored disease really of great importance among this population. The African American community is not particularly aware that they have an increased susceptibility to this disease, and care providers focus a lot on the increased susceptibility among Asians, but I would say are generally ignorant about the fact that African Americans also have a significantly increased susceptibility. And you see the data from the only existing studies that have been published on this slide, showing in red the increased relative risk among African American children. These are our data here from San Diego, where we have a very small African American population, so just the base of children is very small from which we draw our patients. You can see in yellow highlighted the African American attack rate. So this is KD cases per 100,000 children less than 5 years of age, because Kawasaki disease uniquely affects very young children. And you can see that in almost any year that you want to choose, the yellow bar is higher than the attack rate for our Caucasian or Hispanic populations. Having said that, we have a small number of African American Kawasaki disease children here in our San Diego area, and thus the necessity to collaborate with the group at Emory in Atlanta. And this is an example of the data that they collected as part of preparation for applying for this grant. They hadn't really looked at their ethnicity of their patients until we asked them to join us. And they were actually quite surprised to find out that roughly 50% in most years of their total Kawasaki disease population are African American patients. So the way this study is going to run, from my point of view, is that we're going to be enrolling subjects in Atlanta, where the grant is funding a clinical coordinator. We're also going to set up their REDCap database. One of the fabulous things that happened here at UCSD as part of the first biologic demonstration project was that the iDASH group set up a data management system for us, which now is actually going to be shared with investigators in Japan and now with the investigators at Emory. So they've had no system for systematically collecting data on their KD patients, so they're going to be able to use all of our screenshots and just get up and running very, very quickly with that. We're going to take our 60 African American DNA trios here. By trios, I mean the patient and their biologic parents. So we've collected DNA from trios all along. And we've also had participation from collaborators in Chicago and Boston, funded by NHLBI over the last 10 years, who have been sending their samples to us. So we're going to take the 60 African American trios that we have at the Genome Institute in Singapore, where we've been part of an international collaboration on Kawasaki disease. They have 95 trios of African Caribbean descent that were collected in France and the DNA sent to them. So they're going to be doing all the genotyping there. And from the group of patients that we have here, we're going to be collecting demographic data that will also be available from the Atlanta patients. And we'll collect the data that you see there. I wanted to say a word about how these samples were collected from the point of view of the American samples. And we try to cover the privacy issues both in our HIPAA authorization as well as in our informed consent. And I've just underlined maybe the most relevant parts of these consent forms. So we let them know that by signing on to this study, we're creating a database of informations and specimens, such as blood, tissue, and other bodily fluids that may be used in future research, and that this is going to go into a biorepository, a data bank, and a specimen bank. And in the consent form, they specifically sign up for additional research related to Kawasaki disease, so maybe not just the study about a diagnostic test or something that they may have signed up for, but that is going to be used for additional research, that we're going to keep the samples for an unlimited period of time. And this is really important for people doing informed consent who sometimes want to put in, we're going to hold on to it for 25 years or until the study's done, which really puts the onus on the investigator to then make sure those samples are destroyed. And we want to collect things in perpetuity. When I retire, I hope that the bank is still there and future researchers can use it and it can be shared. And we specifically put that in the consent, that it's going to be shared with other investigators at other institutions. And then we have what's called the Moore Clause here at UC, in the UC system, because of a very famous lawsuit and from Mr. Moore. And we always add in that if we were to share this with a company or outside entity, that these samples could have significant therapeutic or commercial value, and that the donor of the samples is giving consent, understanding that they will not receive any benefits from that. And this is required for all consent forms that go through the UC system, system-wide. So in terms of the analysis, are we really going to have enough samples to be able to learn something from the genetics? We think so, and the sample size considerations are given here. We're using the Illumina 650K chip, which will be, all the samples will be genotyped in Singapore. 250 African American and Afro-Caribbean samples is what our target collection is. And obviously we'll have some limitations of power, but you can see the minor allele frequency there. We'll have greater than 80% power if the minor allele is represented at least 5% in the population, and confers an odds ratio of 2.5 or greater. So we won't be able to find very rare variants, but we will have some power. So I'm going to turn it over to Jihoon now, who's really going to address the issues of how we're going to send information. Sending the DNA is easy. It's sending the information that's complicated to be able to share it and come out at the other end with an analytic answer that's important to our science. So I'm going to turn it over to Jihoon.
Thank you, Jane. So I'm going to talk about aim 2 and 3. So two words I remember from the previous talk from Alice is one is privacy as an enabler for research, and also there's operation versus the clinic versus research. So I hope this is aligned with that, talking about good use case of research, and also how privacy is enabler for the general knowledge extraction. So our goal is to develop an approach to construct integrate genotype data from horizontally partitioned data set in a privacy-preserving manner. There's a data partition, there's two things, vertically partitioned or horizontally partitioned. So if you think about Excel spreadsheet, horizontal meaning adding more rows versus where a vertical partition meaning adding more features, variables, more columns. So here we fix the columns, but we are adding more rows or records from different institutions. So two methods I will explain in later slides. That's SMC, Secure Multiparty Computation, and Virtual Machine. So I start with the word, the TDT test, short for a transmission disequilibrium test. This is one of the two common tests in linkage analysis. The other one is association study, where association is simply case control, like affected versus unaffected, or new drug versus placebo, or treated versus untreated. This is a new statistical method to overcome the sample size limitation by using a case and its parent the mom and dads of trios. So unit of analysis is a transmission from each parent. So it's like you can think of a gamete competition from what you inherit from mom or dad, and hopefully that variant is tied to the susceptibility of certain disease or any phenotype you're interested. So strong positivity or extremely small p-value, which suggests that tested marker is a disease predisposing gene or closely linked to such a gene. And this is, statistically speaking, this is another way of calling it McNamara test for matched pair in a 2x2 contiguous table. I will show the details. This is for another slide for non-math version. Jane insisted. This is from the heterozygous parent, child inherits allele A or B, depending on what you get. Hopefully you test for which one is a more positive effect on certain disease susceptibility. And this is for math or Greek math formula version. You inherit from mom and dad, typically rectangular for dad, male, or a circle for female. You color the child for affected one. Simply you create a 2x2 table, whatever you inherit, and you get statistic on your right, and then you check it on asymptotical from distribution. That's where you can get a p-value. Sorry, data acquisition, we get, I think Jane already explained this, we send it to San Diego to Singapore for sequencing only, just to make a distinction that this is a data acquisition part which is different from data sharing part. So this is one high-level view of SMC, secure multi-party computation. Originally, this started from so-called millionaire problem when there are 3 or more millionaires. They want to figure out what is the average of their annual income without revealing their individual income because to avoid the tax or whatever. Here, we are in the same situation because especially UK law, that UK data never goes out of UK without any safeguards. So here we have 3 countries or 3 institutions, US, Singapore, and UK. So we start with number 1, where you can think of random noise as a key. So we log in, only whoever has logged in can unlock the whole data. We send a virtual machine. You can think about the real physical machine. We send to the Singapore, and they add their own data on top of their stack, and then they send the same thing to the UK, and then after adding their data on the top of the stack, the UK does the same thing. With given the whole data stack, they add their own UK data, and then finally end it back to the original place. And then UCSD only can unlock in number 5 the data. So AIM3 is a population stratification, which is must-have method to get published paper out of some GWAS related or any genetics related one. So this is to develop a correction algorithm for population and predictive models for susceptibility to KD in privacy preserving manner. So let me explain about what is the PS, the population stratification is. This means there's a presence of systematic difference in allele frequency in between subpopulations due to ancestry or baseline disease prevalence. This is a primary cause of spurious association. It gives you a lot of false positives. So since we have all African related ones, but they are African American, Afro-Caribbean, continental African, so we need to know what are the spurious association, and if there is, we need to get rid of it. So we use PCA, principal component analysis. Simply put, only you need to remember is the method of algorithm of PCA can be summarized as the big the red box. So there's a matrix that can be easily partitioned. This is very similar to the GLORE, the Xiaoqin's talk in the afternoon, and I'm going to show skip. So mathematically, PCA involves only 2 matrices, the V and then sigma. So we can easily partition in mathematical way, and there are already many methods to implement that. So let me give you this slide just to tell you why PCA is helpful. So this is 2D. So PCA is dimension reduction. Given the tens of many number of features, you can reduce number of features, variables into 2. So PC1, PC2. So given the genotypes, they reduce into 2 variables from the hundred of variables. And then this particular paper, Nature paper in 2008, they compared this data-driven map versus actual map of Europe. As you can see, they pretty high agreement between the genotype map versus actual geological map. As you can see, the Switzerland, the CH is in the center, and the France and left of it, and all the way right on the top there's a Russia, RU, and on the right bottom you can see the other Southern European countries. So this is if you get away from certain, if you're not clustered in a certain group, then that means you are biased from the certain population. So this is how KD samples we applied in our previous publication. We used the PCA to remove certain biased samples. So, thank you. I'll take some questions and comments. Yes.
I'm sorry, from your PCA, you were starting with what set of features?
The genotypes or allele frequencies, yes, and that's
So
all
how many alleles per person?
Human genome has 3 billion.
You weren't using all 3 billion.
Sites, but we use Illumina chip, which is about coverage is about 500 million or 600 million.
Thanks. And you went down to 2 dimensions.
We don't know. That was for publication purpose, but we usually end up with until you the coverage explains like 80% or 90% of data. Yes.
Can you explain any of this in terms of eigenvalue gaps?
Sorry, I didn't hear.
Can you explain any of this in terms of eigenvalue gaps, like the gap between the k and the k plus first is?
Yes. So so how many PCs do we need to get? That's determined by the how far I should go until the number of PCs explain the whole variability, because number of PCs means it captures variability. Adding more PCs means you can add more variability, more explanatory power. But if 4 PCs explain already 90% of data, you don't need to add more PCs. So there's a certain point. There's a lot of method to select the optimal number of PCs. Yes. Yes.
In each country. So then finally sent to UCSD.
Yes.
So does you have all the data, or I just don't quite understand.
Yeah, sorry, I didn't explain clearly about virtual machine because we wanted to explain it in separate section. So you can think of a virtual machine as the player.
I think what was missing is not the data themselves that are being transmitted, but the results of the computation on the data. For example, coefficients that were transmitted from one institution to the other, and then they add their own, and then at the end you recover a final set without having even disclosed the coefficients from your own institution. So, but not necessarily the data themselves. I mean, that it could be that, but it could be a variety of other things or aggregate results.
And for the secure multi-party computation, for the SMC part, I think this is depend on the data, right?
Yes.
For genomic data, if the input size is huge, right, the circuit is going to be very large. So, you have to build up a circuit, to obfuscate a circuit to that party, right? So, if input size is huge, then the circuit is very big. So, have you guys tried these things before?
So when we talk about big data, the definition of big varies. So actually what we each TDT test or any association test is done per variable or earlier site. So we don't need to send the whole data as a set. We just need to send it per sequentially, each side by side.